DMS is a nucleic acid modifying chemical, so before you begin, be sure that you are working in a fume hood with good air ventilation and are wearing double gloves so that even if DMS comes into contact with one glove, you can remove it and still be protected. It's also helpful to have extra gloves available, and you want to have the appropriate waste containers, including liquid waste, solid waste, and a sharps waste container. You should also be wearing a lab coat and safety glasses. DMS is stored in a sealed container, and when you are taking DMS out of the container, you want to be sure that you do not create back pressure. So you can use a balloon filled with nitrogen attached to a needle and insert that into the seal before you then extract the DMS with a needle on a syringe. Add your plants to the buffer. We are showing you this protocol using plants, but it can be easily modified to other organisms. Microorganisms or cell lines grown in liquid culture would not have this step, as DMS can be added directly to the culture media. And once you add your organism of interest to a buffer, be sure that there's enough buffer in there that it will completely cover the plants when they're placed in the buffer. Remove DMS using a filter pipette tip. Work quickly once the organism is in the buffer to minimize additional stress. Here is where DMS can be added directly to liquid culture of certain organisms. Add the DMS to the organism in the buffer, disposing of the pipette tip in the appropriate waste. Start the timer for the appropriate time and shake the tube to ensure proper mixing and minimize air bubbles on the organism. Continue shaking or inverting approximately every 30 seconds. When the timer is finished, add DTT to the appropriate concentration and shake this also to dissolve solid DTT. This reaction proceeds for 2 minutes. After the 2 minutes, dump the buffer into your liquid waste container. The following steps may be different for microorganisms or cell lines grown in liquid culture. Dispose of culture media, rinse, and freeze as appropriate. And add fresh distilled water to rinse. Even though DMS should be quenched by DTT at this point, care must still be taken. Shake a little to rinse. Decant into the waste container and repeat a second time. After the second rinse, dump out onto a paper towel. Again, work quickly to prevent RNA degradation and blot to dry as completely as possible. Then place into a new Falcon tube. Handle carefully at this step. One may consider using forceps. And then freeze into liquid nitrogen. This procedure must also be repeated with a no DMS control, but still adding DTT. All material must be disposed of in the proper waste containers.